everyone. Welcome to Chapter 18, Part 1. So in this lecture, we're going to start with the endocrine system. We will, my plan is to go all the way down to the chemical classifications of hormones, ending here with this slide. And then what we'll do is pick up with, with this slide, and then we're going to pick up in the next lecture with factors affecting hormones, things like that, first, second messenger systems, how they act differently. So what we want to start with is, as with always, is our little quote that I've got with every chapter. And this one I chose was, success isn't owned, it's leased, and rent is due every day. So just remember, you're always putting in the effort for your success. It is your future that you're investing in. So as we go through the material, remember that when I write my exams, I'm going to be asking stuff on each and every one of these objectives. So the difference between an endocrine versus exocrine gland, stuff like that. I'm not going to read them to you, as you can see this, and it's not something that I'm not going to insult your intelligence by reading to you. But what I do want to get us started with is really just a quick recap of something that we learned in A&P 1. And that is the idea of feedback, positive versus negative feedback systems. So with positive and negative feedback systems, I am using a slightly different method for drawing. And sorry, I just kicked my recording table here. Okay, so what I want to start going into is the difference in positive and negative feedback. Now, when we deal with feedback, remember, we talked back in Chapter 1 about the concept called homeostasis, the constant internal steady state, that we are capable of maintaining an internal steady state within cells, within living systems. Now, with that, this requires feedback. Now, feedback can basically be broken down into two different groups. Now, if I have the homeostatic range of something being controlled, let's say, for example, blood glucose levels, blood pressure, sodium levels, whatever, and these two lines represented the homeostatic range of those things, then what can happen is ultimately you can have the change in these things. So what we're going to do on both of these is we are going to do both of these either having an increase or a decrease away from homeostasis. And what we are going to do is call these two on each the stimulus. Remember, stimulus is the change in the level of something being regulated. Now, body's response can do two things. Now, what I'm going to do is we can have one response where the body responds to bring things back to homeostasis. BR, body response. And also here as well, BR, body's response. For example, the stimulus could be an increase in blood glucose, where the body responds to release insulin to lower glucose back to its homeostatic range. Whereas here, the stimulus could be glucose levels dropping. Body responds by releasing glucagon to raise it back to its homeostatic range as well. Everything has that range. And we call this type of feedback because it is always negating the stimulus. The stimulus and the body's response always go opposite, is negative feedback. Okay? Negative feedback. Now, a negative feedback, remember, the stimulus and the body's response always go opposite. Now, in this one, I'm going to use red. Let's say the stimulus, something went up, something in the body increased. The body could then respond to make things worse in both directions here. Especially here. Okay? Especially this one. This is the one that we see when we deal with it, like childbirth, things like that. When we deal with countercurrent multiplication system in the kidney. So things like that is the stimulus and the body's response go in the same directions. This always takes us away from homeostasis, increasing the disturbance, pushes us to completion. So we would say this pushes, so it's positive feedback. Now, with the two types of feedback systems, remember that it is always relating here the stimulus and the body response to each other, where the stimulus and body's responses are going in relation to each other. Okay? Now, as we go back to that, remember diseases come by disturbing homeostasis. A lot of your diseases, like you reabsorb too much water, you can end up with constipation. Reabsorb too little water, you can end up with diarrhea if you're doing in the digestive system, for example. So these can be loads of endocrine diseases: diabetes mellitus, diabetes insipidus, Cushing's disease, Addison's disease, some of the ones that I talk about in this chapter. So as we go through, we also have to understand something really big. And I'm going to talk about this a little bit, but we want to make sure we first understand what the chemicals here are that are controlling the body, called hormones. That is the thing, the chemical messenger, the chemical controller used by our endocrine system. Remember, the system of glands that secrete hormones was the endocrine system. And it goes to a cell that can respond. Now, there can be a cell or cells. For example, we talked in AMP1 that calcitonin, for example, could impact the osteoblasts and the osteoclasts in two different antagonistic ways. And those were both target cells. So the cell that responds to the hormone will be secreted, and the cell that responds to said hormone is referred to as an target cell, Now, what I do want to talk about is how this mechanism goes about, what is happening here, and let's really kind of sketch this and talk about, let's say I have a gland here, okay? And this gland, I'm going to draw it very fluffy to kind of give us a classic endocrine gland look to it. And an endocrine gland, and then over here, let's put its target cell all the way over here. Now, we have to be able to get from endocrine gland to target cell. How are we going to do that? Well, you are going to need a system called the circulatory system, which we use a circulatory medium called blood that can help get that material through the body, blood. Now, this blood, as we know, is found within blood vessels, and this is a fluid that circulates many things. We'll talk about blood on a separate chapter. But what is going to happen is you have a fluid around this. This is a type of extracellular fluid, oftentimes called the interstitium. You have extracellular fluids. And what is going to happen, this endocrine gland here, because it is a ductless gland, that it lacks any kind of duct, it will secrete chemical messengers called hormones into this ECF, into the extracellular space, the fluids, and it will secrete there, and then these will diffuse into capillaries, and the blood flow, let's say blood was traveling in this direction, okay, let's say this was the direction of blood flow here, and we were able to trace that blood flow. We're going to see these hormones will enter the blood, be carried by the blood, and when we get to capillaries, we can eject those hormones out. And when this hormone here, let's say, leaves, the bloodstream, then what we're going to have on this target cell is a receptor protein that, for example, can interact with said hormone here, and it's going to have a shape that allows it to interact. Chemically, these guys will have this kind of fit. The hormone will have a shape compatible to said receptor, and because of the hormone shape, it can bind to the receptor, and what it will do is this receptor here will, because of the hormone's actions, will either be turned on or turned off. You can take this target cell and decrease its metabolic processes, or you can increase its metabolic processes. There's a variety of things you can do with that. You can increase, for example, enzyme secretion, production. You could turn down cell division, turn up cell division, turn on and turn down the protein production. A variety of impacts. Adjust this cell's permeability to certain molecules, things like that. Okay. So a slew of metabolic alterations can happen because of that. So what happens, as I said, this endocrine gland makes a hormone, and it goes into the extracellular fluids, the extracellular space around my cells, around the secretory cells of the gland. Because these cells and these glands have no duct, then from that extracellular fluid or space, we go into the blood vessels, we travel with the blood flow, and it will arrive to a target cell and affect that target cell's functionality. Have a big impact to either stimulate it or inhibit its various metabolic activities. Now, remember there were two kinds of glands. Now, we talked about this in histology section of A&P1, where we talked about the endocrine glands and the exocrine glands. And we said endocrine was ductless, lacked ducts, has no duct, and exocrine does have a duct. Now, endocrine glands, because they have no ducts, remember, they secrete into the extracellular space or extracellular fluids. That bit gets picked up by the bloodstream. Hormones go to the bloodstream. So hormones are made by endocrine glands. Exocrine glands with ducts, they don't make hormones, they make other secretions. They tend to be things like that go to the body surfaces or into the cavities. For example, the serous secretions of your serous membranes, the transudate, the serous fluid, sweat, oils, sweat glands, sebaceous glands, mucus glands, mammary glands, glands we talked about in A&P1 in the skin, and things like that. We talked about some of these cells, and then mucus glands I'll talk more about later on in the respiratory and digestive system. But these go into like body cavities, things like that, and body surfaces, okay? But remember, endocrine, duct is not present. No duct in endocrine. Exocrine has ducts, secretes into those ducts. The ducts are utilized. Now, I am giving a list here, and right now I'm just kind of going to familiarize you with these names. Lab is doing this as well, or actually has done this as well, for those of you guys who are watching this in the summer. But the hypothalamus, the pituitary, which is going to be the anterior and posterior lobe, the pineal, the thyroid, your parathyroid, your thymus, the suprarenal or adrenal glands. Now, you will hear me say suprarenal most of the time because I find that more descriptive. The pancreas and the gonads. Okay, gonads include testes and ovaries, depending on your biological development. Even necessarily, you can have issues with duct regression, though one has a certain biological sex. So, now, what we'll talk about with the gonads, testes, and ovaries. Now, as I do like to talk about things like our hypothalamus is up here in our brain. It's actually where the third ventricle would be. Here be the thalamus, and below the thalamus is the hypothalamus. At the back here, part of the epithalamus is the pineal gland, our pituitary gland right here below the hypothalamus. Then as we go to the neck, you will have the thyroid, and then at the back, the two on usually two on each lobe. For most people, that's not all people, but you have the parathyroid glands. Just here overlying the heart, behind the sternum is the thymus. Then on top of the kidneys will be the adrenal glands. Wrapped up behind the stomach a little bit here is our pancreas. And then in this, the male, the gonad, testes, in the scrotum, in females, a little bit higher up, the ovaries. And we'll talk about what these guys secrete, what they do, and the basics behind them. Now, the next big thing, and I do tell my students for this course that at least 6 points on the test, at least, that's 3 questions, is matching glands to their secretions. Now, we did this in lab, and we do it in lecture as well, so you guys do get it twice with me throughout the semester. But one of the things you will need to be able to do is to match secretions to the gland. For example, the hypothalamus makes antidiuretic hormone, ADH. Oxytocin, OT. RH stands for releasing hormones, and IH stands for inhibiting hormones. These will be hormones that are known to regulate the anterior lobe of the pituitary and its secretion, okay, is the RH and IH. Now, the anterior pituitary gland has 2 separate lobes. 2 separate lobes of it. One is called the anterior pituitary. Now, anterior pituitary makes ACTH. What is that? It's adrenocorticotropic hormone. FSH, follicle stimulating hormone. LH, luteinizing hormone. PRL, prolactin. TSH, thyroid stimulating hormone. MSH, melanocyte stimulating hormone. And GH, growth hormone. Now, look at the posterior pituitary and make a very big note here. You will notice that oxytocin, OTOXT, and ADH are both listed here. They are listed here and here. Now, why is this? What is going on? Well, first off, you guys should know from lab already that the hypothalamus produces ADH and oxytocin. But the oxytocin and ADH are stored and released by the posterior pituitary. Why that is there are neurons, neuroendocrine cells that are doing this. Now, thyroid, T3 and T4, okay, that makes it easy. T3, T4 with thyroid. And calcitonin. Now, we'll talk about triiodothyronine and tetraiodothyronine here in a moment. And calcitonin, CT. Parathyroid, PTH, parathyroid hormone. Now, the suprarenal or adrenal gland actually has 2 major areas that secrete. One of those is called the adrenal cortex or suprarenal cortex. Those make what we would say is androgens, mineralocorticoids, and glucocorticoids. Now, remember this. You will see that the named hormones for adrenal cortex begin with an A or a C. Androgens, aldosterone, cortisol. Androgens, your mineralocorticoids. Aldosterone is our main example. Glucocorticoids. Cortisol is our main example. Medulla of the suprarenal gland, we know from the autonomic nervous system, produces epinephrine, norepinephrine. So we know the suprarenal medulla, remember, contains the postganglionic neurons of the sympathetic division that are short. That's the unusual thing about them, is all postganglionic neurons of the sympathetic division of the autonomic nervous system are long, except the suprarenal medulla. Now, the pineal gland makes melatonin. The thymus makes hormones called thymosins. That's pretty easy to remember. The pancreas, we should all know, makes insulin, but we also know it makes glucagon. Now we're going to see a hormone called GHIH, growth hormone inhibiting hormone. PP, pancreatic polypeptide. This is your pancreatic polypeptide. Okay, PP. Now the gonads make sex hormones. If it's testes, primarily testosterone. Now it does make things like inhibin, which I talk about in the reproductive system. Okay, that will be more repro. Estrogens and progesterone. The ovaries make estrogens and progesterone. Now estrogens, it is accurate. There is actually 3 types of estrogen typically made in the female. And we'll talk about those in at the end of the semester in the reproductive system, chapter 28. Now, what I want to make sure we also understand here is this endocrine system works with the nervous system, and they are kind of like very opposites. Remember back in A&P 1, for those who have me in A&P 1, I described them like it was the tortoise and the hare. Remember the story of the tortoise and the hare where they were going to have a race, and the hare, the rabbit, he took off fast as can be, bam, and he was so far ahead of the tortoise, he was slow and steady, that the rabbit, the hare, took a nap, and the tortoise just kept going, and the rabbit kept sleeping. Eventually, the tortoise, who is slow and steady, the rabbit oversleeps, and the tortoise overtakes him, and the rabbit was so far behind, he could never catch up, and he lost the race. And this is kind of like with the endocrine system is the tortoise. Very long-lasting when compared to the nervous system. Remember we talked about the suprarenal medulla pathway. We said that it did 2 things. We said suprarenal medulla allows us to control things where we don't have neurons directly, and also allows us to have sympathetic innervation for a longer period of time. Now, not only that, but our nervous system works alongside of endocrine to maintain homeostasis. For example, the heart rate, the breathing rate, the digestive processes oftentimes are controlled by nervous system. But hormones also affect digestive processes. We will talk that there's neurons, helps control heart rate and contractility. But we also will see that there will be hormones that will affect heart rate and contractility as well. And together, these 2 systems control homeostasis, but they do so very differently. Now, if we did a direct side-by-side comparison like we are doing on this slide, nervous system, we would say it's wired. Why do I say it's wired? Well, when you think about the axons of neurons, like a wire, going from for example, in the somatic motor pathway, you have a lower motor neuron coming from the motor nuclei of the central nervous system, all the way down to the skeletal muscle cell where we have an NMJ. Well, and that has synapses. Well, in the endocrine system, you don't have wires. Now, nervous system uses neurotransmitters, where we in endocrine system are using hormones. Now, what about when it comes to the suprarenal medulla? It is a neurotransmitter, epinephrine, norepinephrine, but it is released in the blood. That makes it a hormone. This is why we can look at it and be like, we can classify it one way when we're discussing neurotransmitter, and another way when we're discussing hormone. Now remember, neurotransmitters always go very short distances across synaptic cleft, not a very long distance. Whereas hormones can go incredibly long distance, traveling through the miles of blood vessels before it actually gets, sometimes miles, before it actually gets to its target cell somewhere in the body. Now, because neurotransmitters diffuse very short distance, the receptor is very close to the synaptic terminal. Over at the postsynaptic membrane, for example, the motor end plate of a neuromuscular junction, or just the postsynaptic membrane of a typical neuron-neuron synapse. But the receptors of endocrine can be very far away from glands, especially when you're talking about I release it and it's got to go through miles of blood vessels before it ever gets there. But it could be on the other side of the body. Now, one thing about nervous system, the commands are very quick. It acts very fast response. But endocrine has very delayed responses. There will be, hormones can get released, and it takes time for the response to happen. But this is why I said it's tortoise and the hare. It took a while for the tortoise to really get going, but the tortoise didn't stop. The command for the rabbit, the hare, was very quick to get going, but he quit, took a nap, said, I got so much time, I'm so far ahead, I'm going to take a nap, and still beat the tortoise. And he does. So the commands are very quick for nervous system. They're very delayed in the endocrine system. Nervous system, it's very short duration. In fact, usually it's just and done. It fires, and we do the one command until we're ready to do the other command, maybe very soon after. For example, in MJ, we talked about how the number of times that neurons are stimulating muscle cells can affect the amount of tension over time. So now, one of the things nervous system do is very quick things, like knee-jerk reactions, jumping out of the way of a speeding bus. But endocrine is used to regulate body systems, like going through puberty, development, reproductive growth of gametes, things that can take us a little bit more time. It can take us up to weeks time frame to take a follicle, and it takes about 2 weeks to get a follicle that was already beginning meiosis and let it finish it under the control of hormones. So things that can take 2 weeks, things that can take years, like puberty, hormonal spikes and changes in timing of hormone expression can change the way your body develops. So, as we think about that, regulating body systems, okay? Now, remember, we drew this out right here. This drawing right here basically is doing what we call an endocrine pathway, okay? You always are going to have your endocrine gland going to the blood to a target cell to impact that target cell's metabolism, okay? And that's the basic pathway. An endocrine gland, sorry if you can hear my wife upstairs. An endocrine gland is going to make the hormone, and hold on. Okay. Alright, sorry. So, the endocrine gland makes that hormone, it goes into your blood. That hormone is the messenger. Now, what I really want to make sure you guys understand here is we talked about in Chapter 2 in AP1 about different kinds and various types of chemical bonds. And with these chemical bonds, you can end up with unique shapes. And you remember something I told you never to forget is this idea that form fits function. Form fits function. And when I said that back in Chapter 1, I said form fits function. You'll never get past this. The structure of the hormone, its shape, determines that it can bind to the receptor. Some bind with greater affinity than others. When we talk about how norepinephrine and alpha cells, alpha receptors versus epinephrine. Now, what you will see is these hormones, their shape will allow them to bind to receptors, and when they do, we can tell that cell to upregulate or downregulate, stimulate, inhibit its various activities, like enzyme production, cell division rate, permeability of the cell, various things that cell might do. Now, these hormones, we actually refer to this hormone as first messenger. This will be a big thing in the next lecture, is second messenger. First messenger. Now, most hormones will be out there active, usually for about 2 minutes to an hour, for most hormones. Now, some even a little bit longer, because of the protein effect. But I would say basically 2 minutes to an hour is a good kind of reference range for most hormones to stay active. Now, Why that is enzymes may deactivate it, they may get urinated, they may get broken down by the liver, what we call first-pass metabolism, may remove them, things like that. Now, target cells, that is the end. This is my finish line for the pathway. And what we would say is, guys, we have a response. The response is on the target cell. The target cell has the thing to bind to the hormone. It has the receptor. The receptor receives that hormone binds to it, and the receptor basically is going to interpret. It's going to bind, and because it receives this chemical signal, it will result in a response specific to that hormone. Now, usually you're going to have you can have like more than one hormone binding to a receptor. Sometimes you only have one very specific. It really depends on the chemistry, the cell, how similar the two hormones are. Some are greater affinity than others and things like that. And we can take advantage of this chemical fit in drugs, because a lot of times, because of drugs, we can have a chemical that we make in a lab that is synthetic but has the same shape, or at least enough similarity in shape to do that job. Okay, things like Synthroid, for example. So it's very important that we understand that thing in a clinical setting. You're dealing with chemicals that have a very specific shape that allow them to do their job. We can mimic that, we can create that in a laboratory, and then let it try to do what its job is supposed to do, to mimic the actual hormone that was deficit. Okay. Now, these terms I do want to talk about, especially the first four, because they are very specific to us. The first four are the ones, we only learn them here. Now, I will talk about these. They do love to put these on the common departmental finals, by the way, questions about this stuff. And I do ask something about the various kinds of cell-to-cell intracellular communication where cells talk to each other. Now, we're going to kind of think about this as cells talking, okay? And when cells talk, like people talk, sometimes you only talk to those that are the same type and right there next to you, okay? Maybe you don't get out much, so you really only get to talk to your family. Or maybe you just stay within the same little neighborhood. Things like that. Sometimes you talk to yourself. And sometimes you talk to a lot of people. And these types of cell communications are somewhat like that. Now, direct communication. The first thing is they have to be the same type of cells. The two cells are the same type. For example, two heart muscle cells. Now, what happens is ions or other molecules, it could be ions like sodium ion, can cross between the gap junction of one cell to go to another cell. But this is the same type, like heart muscle contraction. Now, this type of cell communication is very specialized, kind of rare, because this is typically what happens in the heart and places like that. The heart is the only place we're ever really going to discuss it. Now, paracrine. Now, paracrine is by far the most common. This is used in, for example, lymphatic system. Cell to cell within the same tissue, like lymphatic cells, where you have cells in a tissue where they communicate within that single tissue, things like that. A great example of this happens in the pancreas. And if you guys remember me talking about pancreatic islets in lab, and I described the pancreatic islets are like a party where the center of attention in the party is a beautiful woman, the beta cell, and she is surrounded by alpha cells, and the alpha cells and the beta, so the alphas, and the betas have different goals. The alpha cells, they want to raise the sugar.
by making glucagon, and the beta cells want to use up the sugar, making insulin. So when beta cells make insulin, they will release insulin out into the islet, and the surrounding alpha cells are inhibited by this. That is an incredibly important paracrine signaling to really understand in a clinical scenario, as it helps you understand what might be going on with diabetic patient. Now, this is why it's one of the most common, is lots of cells within the same tissue. You can have two, like an alpha and a beta cell, which are in the same tissue, but they're different cells. They use paracrine factors, cytokines, things like that. Like, for example, interleukins, which are between two leukocytes. Like, one of the important things is, I think it's interleukin 6 inhibitor, that was used in the treatment of COVID. I'm pretty sure it was interleukin 6. So I will say on that, don't quote me. I would double check that number. Interleukins is not something I've had to spend a lot of time with on lately because we don't teach at that level. But since immunology, but there are interleukin inhibitors that are used to prevent some of the severe symptoms of things like COVID-19. And autocrine. This is you talk to yourself. The cell makes autocrine factors into the ECF, and that comes back onto the cell. So it's like the cell talks to itself. The cell secretes a signal, a material. It goes out to the ECF and then binds to a receptor on the cell that made it. And so the cell talks to itself. Autocrine. Now, endocrine, we know we're talking to others. We are going to the bloodstream. Okay? Its hormones are in the blood. Remember, they go endo-blood. Endocrine, endo-blood. Into the bloodstream to go to many tissues. You can affect many tissues inside of many organs at the same time simultaneously. You can go affect bone tissue. You can go affect all the bones. Like if you wanted to affect many bones, especially some of the really big ones that tend to get remodeled a lot with calcitonin and parathyroid hormone. Now, synaptic communication, we know, is that synapses, neurotransmitters, remember neuromuscular junctions, neuron to muscle cell, neuron to neuron, neuron to another neuron cell, and neuroglandular junction, neuron to gland cell. Now, neuroglandular junctions can be a way to control endocrine glands. Oftentimes, it's not the only way, but it is a way to control some of these endocrine cells. At least in some endocrine glands, is direct neural stimulation. Also in exocrine cells, a very, very common way. But we'll see other ways. And when we talk about endocrine in just a minute, I believe one of the next things we're going to talk about is, very soon anyway. Well, not, it'll be next class. So as you can see here, direct communication, food gap junctions from cell to cell within the same cell. They have to be both the same type of cell. Paracrine is cell to cell within the same tissue. We use paracrine factors. Autocrine, you go out to the ECF and then you come back to a receptor on your own cell surface. Endocrine goes to the bloodstream, and synaptic always uses neurotransmitters across synapses. Now, autocrines, like autocrine factors, there are various types of autocrine factors. These may be things that, for example, let's say that this cell here makes a hormone, and then this chemical also says, since you made enough, let's inhibit it. And you can act to like shut off its own self as a negative feedback mechanism, a type of feedback inhibition for the cell itself. That now that it's made it, the end result causes a stoppage. So, a variety of functions that it can do. All right. Now, these hormones and these cell signalings, but it's endocrine is our primary focus here. But remember, I could ask any of these, especially direct, paracrine, autocrine, and endocrine. But hormones of the endocrine system, these are some things I want to make sure we focus on at the last part of this lecture. And one of the big things I want to make sure you guys understand is when a cell when a hormone gets, to a target cell, what is going to happen is these 5 things can occur. Okay, I'm going to give some examples when this happens. Okay, so let's say the first thing is we can change cell permeability. Now let's give a great example we're probably familiar enough with is insulin. Now one of the things that's going to happen is insulin is going to make some cells in the body take up sugar, take up glucose, and bring it in. By insulin binding to an insulin-dependent glucose transporter, that glucose is brought into a cell, and we've now altered that cell's permeability to glucose, because glucose is a relatively large and polar molecule. That molecule is going to be able to be absorbed more readily through facilitated diffusion because of the arrival of insulin. Synthesis of proteins. Testosterone, for example, when it goes to muscles and various androgens, the anabolic steroids, we might refer to these two as hormones, they can cause the production of extra muscle protein to increase the size of the muscles and things like that. We can activate and inactivate enzymes. A great example of that was how the osteoclasts on bone tissue would be responding to be activated or inactivated by calcitonin parathyroid hormone. If you activate it through PTH, it's going to release enzymes that eat the bone and release the calcium into the bloodstream. Now we can also have endocrine glands to cause the production of secretions. Now this is something we don't really probably have a big handle on yet. We will later on. One of the biggies I'm going to say is this, is a hormone that we make in the hypothalamus, GnRH. GnRH from the hypothalamus will go down to the anterior pituitary through the hypophyseal portal system, and it's going to cause the release of FSH and LH, or at least the production of FSH and LH. In females, it does not cause the direct release of LH, as we'll see in repro. And we can also stimulate cell division, like mitosis and meiosis. For example, growth hormone can cause increased cell division through mitosis. Follicle-stimulating hormone, FSH, can cause the increase in meiosis and gamete production. So those processes get stimulated by the arrival of those hormones to the gonads, or to like growth hormone to like a bone cell, to where the area, the growth plate we talked about, that those cells can then increase in cell division to grow taller and cause bone growth up till puberty is over and you no longer grow. So, alright, now when these 5 things happen, when the hormones have these 5 actions, these are 8 things that can be done by that, 8 results. Now, you could probably agree, if I do 1, 2, 3, 4, 5, I could ultimately maintain homeostasis to some degree. Okay? To some degree, at least 1, 2, 3, 4, at least, could be definitely done. And we have probably no trouble understanding that if I increase cells permeability, I can lower the level of something in the blood. If I make proteins, I can actually, by producing some proteins, I could be taking chemicals out of the blood that could be doing other things and turning them into proteins and lowering it out of the bloodstream, for example. Even preventing that molecule from being turned into glucose through liver process called glucogenesis. We could regulate metabolism. For example, if you change cell permeability, you're going to let glucose come in. If you're going to activate enzymes and secretions, you could activate different metabolic processes. Okay? Regulating muscle contraction, specifically the autonomic epinephrine, norepinephrine, if it was secreted as a hormone, can come in to cause heart muscle contraction, cause smooth muscle contraction in blood vessels and things like that. Regulating gland secretions. We'll talk about how, again, that certain hormones will cause glands to be secreting other hormones. We can regulate growth. For example, metabolism, giving energy available to grow, regulating cell division to grow, things like that, to regulate development, again, metabolically, growth and development. For example, as you develop, remember with the right hormones, like anti-Müllerian hormone, the Müllerian ducts can regress, things like that. And we'll see those kinds of things happening. And we're even regulating reproduction, like, for example, sex hormones, to help produce sex cells, gametes, to affect sex drive, libido, to affect sexual characteristics, development of a penis, development of a clitoris, development of the proper organs of sexual copulation and their appropriate functions, even to being able to become aroused sexually, to be able to have sexual intercourse. These things must be done and needed to have appropriate glandular secretions to make semen or other sexual fluids, like the female's glands to secrete lubricants into the vaginal entrance, things like that. These were all needed to regulate reproduction. Now, a very big thing that we're going to be getting into is the three major chemical classifications of hormones: amino acids, peptides, and lipids. Okay? Amino acids, peptides, and lipids. Okay, so amino acids, peptides, and lipids. Now, these are the three big chemical classes that are present in hormones. They are derived from amino acids, from peptides or smaller proteins or proteins, and or lipids. Now, this requires us to have some knowledge from chapter 2, the big organic chemistry section. Now, I spend 4 lectures, 2 weeks on chapter 2 in my lecture for that very reason, to teach all the in-depth chemistry, to make sure my students know the details about these. So as we go through them, what I want to talk about with each of these is they are made through different things. As you can see, amino acid derivatives, for example, are built off of amino acids. You can tell this is an amino acid. Here's a carboxyl group. Here's an amino group. Amino group, carboxyl group. COOH, one of the O's is gone off of here to add this catechol group on here. Things like that. Okay, so let's talk about amino acids. Now, to help you guys learn the amino acids, what I want to start off with is there are 2 major groups: catecholamines, thyroid hormones, and melatonin. Okay. So there's one group is the catecholamines. That's the big group. Catecholamines, thyroid hormones, and then good old-fashioned melatonin. Okay, now, catecholamines. That's going to be the big hard one here, okay? Catecholamines. That is correct. Okay, catecholamines. I want you to think, can elephants never die? Can elephants never die? Question mark. Can catecholamines. Elephants. Epinephrine. never, nor epinephrine, and dopamine. These are three very important catecholamines. Now, somebody says, Lefford, why are you adding dopamine here when it's not mentioned in lab and anywhere else? You guys will talk about drugs constantly that affect various catecholamines. And if they affect some catecholamines, they can affect more than one: epinephrine, norepinephrine, and dopamine. Catecholamines end in "ine", but they have, they're actually called catecholamines because they, and the way they get their name, and you do not have to know this, but they have what is called a benzene ring on it that have two hydroxyl OH groups on it. That's where they get their name. That is actually referred to as a catechol. Now, thyroid hormones are 1 and 2. These are T3 and T4. Now, you're wondering, Ledford, thyroid makes calcitonin. Why is that not in here? Calcitonin is a peptide. Thyroid hormones are T3, T4, and melatonin is on its own. So, what we want to do here is make sure we have catecholamines. Can elephants never die? Thyroid hormones, T3, T4, melatonin here. Now, what is very interesting is your catecholamines and your thyroid hormones are all made of tyrosine, an amino acid. Tyrosine, remember, is also used to make melanin. We talked about in AP1, if you don't have the enzyme tyronase, you can't turn tyrosine to melanin, and this causes you to have albinism. And then tryptophan, which is another common amino acid, building block of proteins. Melatonin is made out of that. Some people used to say that's the reason why you got sleepy is all the tryptophan in turkey at Thanksgiving. I was like, no, it's all the carbs that you ate and all the other food that you ate. You have your insulin spike and your parasympathetic nervous system is kicking into gear and you want to go to sleep. So that's the amino acid derivatives. Okay? Now, peptide hormones. Now, the peptide hormones, they exhibit usually as amino acid chains. They could be prohormones, the inactive ones, and they may have a carbohydrate attached to them. Specifically, the glycoproteins. Now, glycoproteins is the first group we're going to talk about, is the glycoproteins. Now, glycoproteins is our first big group. Glycoproteins. Now, I'm usually one of the professors that is the last done in the semester. I am like last day still trying to teach because it seems to take me longer than some of the other professors. And that's okay because I know that great teachers finish last. Okay, so it's alright if I'm the last one done because great teachers finish last. Great glycoproteins. Teachers, TSH. Finish, FSH. Last, LH. Great teachers finish last. Okay? These are very big. They are very big. They are over 200 amino acids. And they all have carbohydrates stuck on them. Okay? And they're all made from anterior pituitary, by the way. Now, the one that's going to be hard to remember is this guy. Now, here's how I have my students remember it. Is I prefer more a great cookie over a green apple, please. So we know that you probably should eat apples, but I prefer a great cookie over a green apple, please. It's like, I'd rather, give me a good old, nice, salted caramel, chunk cookie. Oh, warm and gooey. So that's a good way to help remember them, okay? I will help you guys retain these. So we will fill this out, but we're actually going to use the I prefer more a great cookie than a green apple, please. That's actually courtesy of one of my former students who is going in. Actually, I need to see how Elena is doing. She should be starting nursing school this fall. If not, I'll have to contact. I need to reach out, see how she's doing. So we will fill this one out in a minute. Is short polypeptides. And there's a bunch of them, the short polypeptides. And this is the hard one to remember, so we'll fill him out last because I'm going to use my mnemonic. Short polypeptides. Now, lipids. Lipids fall under two things. Now, first off, I'm going to do something here. I want to make sure you guys understand something here for the most part. For the most part, anyway. We're going to see that amino acids typically love water, and peptide hormones typically love water. But lipids, they don't love water because love hurts, love scars, takes a lot of pain. Okay, so they do not love water, okay? Lipids are hydrophobic. They don't love water, okay? Now, lipids, hormones anyway, come in two major types here. They're going to be steroids or eicosanoids. Now, steroids, number one, steroids. Now, steroids, if somebody ever this is a way to remember it, this is not life advice. But if somebody offers you steroids, you should accept. You should accept. I'll take your steroids. ACCEPT. Find out what it means to me. Okay, we want to accept. Now, accept, what we can talk about here is your things like aldosterone, cortisol, calcitriol, estrogens, progesterone, and testosterone. Now, many of them have ON. ON, usually, this is going to tell you you're a steroid, okay? And then, so all is telling me it is, but ON is steroid. Now, the second one is called the eicosanoids. Eicosanoids. And eicosanoids are falling under L and P. Okay? L and P. I want you guys to think eicosanoids to me sounds like a hipster band that only releases their music on vinyl LP. You have the leukotrienes. Remember, eicosanoids are leukotrienes. Remember, those are, they have, their fatty acid tails have 3 double covalent bonds in them, and they're made by white blood cells, hence leukotriene. And then lastly is a prostaglandin, which we know prostaglandins have a 5-membered ring and 2 fatty acid tails on it. And these 2 fatty acid tails, things like that, these make them lipids. And leukotrienes have fatty acid tails with at least 3 double bonds in them. And these guys, remember, prostaglandins are involved in pain and inflammation. We talked about how I always was telling my students in AP1 about prostaglandins and they're involved in pain and inflammation. I said if sometimes that somebody goes to visit a prostitute, they may end up later on having pain and inflammation. Okay. Now, what I want to help you guys understand is that steroids are made from cholesterol. Remember, they have 4 fused rings. We talked about this in AP1, that they have 4 fused rings, where these rings that they have fuse together in this particular structure, where there are what we refer to as 3 6-carbon rings and 1 5, or 3 6-membered rings, 1 5-membered ring. And that's your typical steroid skeleton that we have right here. Okay? So remember, with steroids, you should accept aldosterone, cortisol, calcitriol, estrogens, progesterone, and testosterone. Eicosanoids release their music on vinyl LP, leukotrienes, and prostaglandins. And there will be multiple questions. I'm going to go say here's how I do it. In my exams is there will be an amino acid, a peptide, and a lipid. Okay? Which of these is a catecholamine? Which of these is a thyroid hormone? Which of these is a short polypeptide? Things like that. What I'm going to do here is I'm going to do this game. I'm going to do this just so because I am not used the I prefer more a great cookie mnemonic before. So I prefer more a great cookie over a green apple, please. All right. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. All right. Now, these are really good ones to use. I prefer more a great cookie over a green apple, please. Now, insulin. Now, with P, a lot of the times, now you can use, we have prolactin, and we also have PTH. You can use either one of these. Let's just use PRL, MSH, A. Now we have 2. You have ADH and ACTH. G, growth hormone. Now C, we have calcitonin. O, oxytocin, OT or OXT. OXT is primarily used more like in academia. Like in my world, OT is more likely to be used in healthcare. ACTH, growth hormone, and then we have growth hormone inhibiting hormone. ADH, ACTH, and then, I bet, what happened is, okay. I prefer more a great cookie over a green apple, please. Okay, I see what she did. It's pretty good. It doesn't get the natriuretic peptide. And the A of this one should be lowercase and actually not. So I do apologize. I was using it because it was one of the best ones. And I'm going to put at the very end here is your natriuretic peptides. So sorry about that. It's well, here, let's do this. It's my fault, and I do apologize for not catching that in my last edit of my notes. Something that does happen sometimes. Okay, and that'll be natriuretic peptides, and then PTH. Okay. And that was about as good as I could find there. I had a horrible one, and I did like hers better, and then I just didn't realize when I added hers to it. But it does get most of them, and it is a really good one. So I still will recommend to get the majority of short polypeptides by using, I prefer more a great cookie over a green apple, please. It's just that this right here, oh, I bet she was doing atrial natriuretic peptide. That's probably what she did, and I'm just not thinking. Okay. That's not the only natriuretic peptide, and I was just doing natriuretic peptides. I think that's what she did. I do apologize. This is my very first semester using this mnemonic device made by a former student of mine in a previous semester. So I was using it, and that's actually, I had a little contest, and everybody got extra points for making it, but the one that I liked the best would go in, and this is the one I liked the best. It was easiest for me to remember. This is only my first time using it, so I do apologize for my little hiccup there, but I do believe that she was just thinking atrial natriuretic peptide, but there's also brain natriuretic peptide, ANP and BNP. So I was just putting NP for natriuretic peptides, and I do believe that she was ANP, and I do like it. It's a really good one. I do like that one, and I think I'm going to keep it, and I'm just going to maybe do this just as a postscript note, just for future reference, just so I have that. Okay. And the vinyl LP eicosanoids. All right. So this should be where we end this first lecture. Guys, this should be all of it. I will have this up on YouTube very soon as well. But thank you so much for watching. I hope you really learned a lot. I do test on these. And so if you have something, this is a thing that, as you can see, I can erase it. I actually have the ability to wipe it clean with a clean thing and reuse it. It's something I've been using a lot to reduce my paper. And also, you can back everything up you do on here by scanning it with Rocket Notebook. So if you're somebody who wants to look and feel with pen and paper, but is wanting to be ecologically conscious and use less, I recommend it. And I've got several notes that I'm working on and things I'm doing in it for my own personal edification as well. So guys, this concludes my lecture. I really hope you learned a lot, but thank you so much for watching, and as always, I'll see you in the next lecture, guys. Thank you so much.